Today we are going to take the second part of our 7th chapter, Genetics of the Future. Last class or previous classes we were studying about the topic genetics. In our 6th chapter we studied that the father of genetics, that is Gregor John Mental. He found out that he made so many experiments with the pea plants, and he found out that a factor is responsible for the expression of different characters in the offspring. And he also told that the characters that the offspring get from their parents or transmitted from the parents to offspring. The characters that is transmitted from the parent to offspring is known as heredity. And those characters that are different from their parents, that is present in the offspring, is known as the variation. So all these things we have studied. And so gene is responsible. You heard that, or we studied that gene is responsible for the different characters present in our body, and our fathers and mothers or parents' characters are transmitted through the genome to the offspring. Genome means sex cells, and this sex cells, their fusion, fertilization, all these things you have studied. And the reason for the variation also you have studied in the previous class, that is crossing over, mutation, fertilization. All these are the reasons for the variation. Is it clear for you? So variation means what? The characters that is present in the offspring that are different from their parents. That is the variation. Then we have studied about genetic engineering. So with the help of this genetic engineering, we can produce the desirable, an organism with the desirable trait. We can change the character of an organism by changing the genetic constitution of that organism. So, for example, for that, in the previous class, we have studied about the insulin-producing bacteria. Normally, bacteria cannot produce insulin. So, but we have done with the help of the genetic engineering. The basis of genetic engineering is cutting and joining the gene. So, the non-functional gene is cut with the use of, with the help of an enzyme called restriction endonuclease. And instead of that gene, we are placing another gene that is from the human. The insulin producing gene is cut using the restriction endonuclease enzyme, and from the bacteria also a gene is removed with the help of this restriction endonuclease, and with the help of genetic glue, that is ligase enzyme. Ligase is known as the genetic glue. So with the help of this genetic glue, we are joining the gene, insulin producing gene that we have cut from the human DNA. We are joining that insulin producing gene to the DNA of the bacteria. Is it clear for you? So in the bacteria also, what is the genetic material? DNA is present, and on the DNA what are present? Genes are present. So we are cutting the gene from the insulin producing gene from the human, and we are human DNA, and we are placing, joining with the help of enzyme ligase, we are joining that gene on the bacterial DNA, and it's also known as plasmid. And under the laboratory condition, that bacteria is allowed to culture or multiply. And when it multiplies, it will produce what? The next generation of that bacteria will get what? The next generation is also having a genetic constitution. Insulin producing gene is present in the next generation of that bacteria. So this bacteria will produce under the under the laboratory condition. This bacteria is capable of producing inactive insulin. When this inactive insulin is produced, we will convert that into active insulin. This is clear for you. And about the insulin, we have already studied about the function of insulin. Insulin is a hormone. We have studied in our third chapter about the function of insulin. That is, insulin regulates the glucose level in the blood. So, if the glucose level in the blood is increased, and in a person normally producing insulin is not functioning, then what's happening? If a person is highly diabetic, has to take insulin injection. In that case, artificial insulin is given. For all these purposes, we are producing artificial insulin. And how we can produce artificial insulin, one of the methods that I have told you about is the bacteria, with the help of the bacteria. This is clear for you. So that's one of the thing about the genetic engineering. Next, about the gene therapy. Gene therapy means I have told you we can cut the gene and replace another gene. So in the case of genetical diseases, so many genetic diseases are nowadays. We are hearing about hemophilia, sickle cell anemia. All these are genetic diseases. Likewise, so many other genetic diseases are also there. So in order to remove such genetical diseases, science has found out, as science has developed or as technology has developed, this gene therapy has a great role. As genetic engineering has arrived, this gene therapy is one of the treatment method. That means the disease causing gene, there is a gene and there will be a gene which is non-functional or defect in the gene. So that disease causing gene can be cut using the genetic scissor restriction endonuclease, and we can replace another gene instead of that disease causing gene. So that if that disease causing gene is removed and another gene functioning, another normal functioning gene is placed there, what happened? That person will be, or his next generation will be free from the genetic disease. Otherwise, that then genetic diseases will be transmitted from one generation to another generation. Is it clear for you? So that's why some of the diseases, hereditary diseases, we call it as hereditary diseases. That means if our grandparents is having some genetic diseases, that will be transmitted to us also. Is it clear for you? So these diseases can be cleared. One of the methods for clearing these diseases is gene therapy. In gene therapy, the disease causing gene is cut and removed, and instead of that, a normal functioning gene is replaced with it. For cutting the gene, we have already studied genetic scissors. It's the enzyme restriction endonuclease, and with the help of the enzyme ligase, we are replacing the normal functioning gene there. That is the method known as gene therapy. Next topic we are going to study is about the Human Genome Project. So I have told you that we can cut the gene and replace another functioning gene. But the problem is not only the countable numbers of genes are present in our body. In a single cell, now so many genes are present. And I have told you, in the center of the cell, nucleus is present. In the nucleus, chromosomes are present. On the chromosomes, DNA is present. On the DNA, genes are present. So, so many genes are present on the DNA. And we are not able to know which gene is disease causing, or which gene should be cut and removed, or which gene is insulin producing gene. We are not able to know. So about 1990s, in 1990s, there was no idea about the position of the gene. So from it started the Human Genome Project to find out the position of the gene. So about around 2003, it was found out that the new project, or with the new method known as gene mapping, we are able to know the position of the gene. The location of the gene is found out with the help of Human Genome Project or gene mapping. So with the gene mapping, we are able to know the position of each gene inside our cell. So when we are able to know each position of the gene, it's easy for us to cut the gene and replace another functioning gene there. Now we can see some points regarding the Human Genome Project. Here you can see the points regarding Human Genome Project. So in the Human Genome Project, some of the points coming is Human Genome has about 24,000 functional genes. So inside our body, in the cell, on the DNA, in the center chromosome, on the chromosome, the 1,000 functional genes are present. Then human DNA includes that there is only 0.2%. You know that our fingerprint is not same. Even identical twins having different DNA structure, and different DNA is present. So in that different each person. Then about 200 genes in human genome are about 200 genes in our DNA is identical, same as in the case of bacteria. I think it's clear for you about the points that is coming under the Human Genome Project. With the help of this Human Genome Project, gene mapping came, and we were about to know about the position of each gene and the DNA. Is it clear for you? Next topic is genetically modified animals and crops. So genetic engineering has a great role and has many useful things can be done with the help of this genetic engineering. And among that is the production of modified animals and crops. How we can produce these modified animals and crops? How we can produce that means the modified animals and crops are produced with the help of genetic engineering. That is, we can remove the gene that is not useful for that organism. So we can remove that gene, and instead of that, we can place a functional, normally functioning or high yield giving, high productivity giving gene can be replaced with it, so that the productivity of that plant or that animal can be increased. Is it clear for you? That's about the highly producing plants and animals. And we can see some points regarding that, how we can modify about the genetically modified plants and animals. We can see some points now. So here you can see the protein required genetic engineering. We can produce the protein required for that. Interferons. Interferon is the protein produced several diseases. Then next protein required, or the not only the protein, and that is for the treatment of disease diabetes. Then endorphin, and that is another protein, and that is produced for the pain. Then somatotropin, and it's a hormone. Somatotropin hormone is produced for the treatment of growth disorders. So interferons are produced for viral diseases, treatment of the viral diseases. Then insulin for the diabetes. Then endorphin for removal of pain. Then somatotropin, that is growth disorders. These are the proteins and hormones we can produce, artificially produce for the treatment of various diseases. And various crops, hybridization, I think you have heard about hybridization. We have studied in our 6th chapter about hybridization. Just the term you have heard. In the hybridization, genetically modified crops are produced according to our need. So according to our wish or according to our desire, genetically modified crops are produced with the help of genetic engineering. Not only crops, animals are also, their genetic constitution is changed with the help of genetic engineering. It's clear for you. Then about the DNA fingerprinting. DNA fingerprinting is found out with the help of the scientist known as Alex Jeffrey in 1984. So DNA fingerprinting or DNA testing, the scientist Alex Jeffrey in 1984 paved the way, paved the pathway for the finding of this DNA testing or DNA fingerprinting. We have already told, or I have already told you that, but fingerprints of even identical twins are different. You all know that. So likewise, the DNA also, the Human Genome Project, there came a point that is about 0.2% differences. Among the human DNA. So even here, the DNA is the arrangement of nucleotides in the DNA of each person is different. Only our relatives, our close relatives, that is our father and mother, are having some similarities in the arrangement of nucleotides. That's why if a child is lost by any natural calamities or by war or by any thing, if a child is lost, after many years, they are getting that child, and their parents want to identify whether it's their child itself. And for such cases, what is done? DNA fingerprinting or DNA testing is done. We are able to know that the child will be having close similarity with the DNA of his parents. So by that testing, we can find. Then another thing is suspected culprit. If a robbery or a murder is done, and from that place, if that robbery occurred place, from the if the police is getting a nail or a hair or a fingerprint, they are able to find and they will get the DNA and result, and they can match it with the suspected person. So by that, the thief, real thief or the real culprit can be found out. Is it clear for you? So keep in mind that the arrangement of nucleotides present in the DNA is different for each person, and only close relatives are having the similarity. Having some similarity in the arrangement of the nucleotides. So that's why the DNA testing has got the name DNA fingerprinting, because like the DNA arrangement of nucleotides and the DNA, fingerprints are also differ from each person. Is it clear for you? The next is about the misuse of genetic engineering. So many uses we have discussed about the genetic engineering, like the use, as we have every time we tell about a coin, that is like the two sides of a coin. Each and everything will be having a use and misuse. So here, our genetic engineering. And the use is we can produce highly modified crops. We can use it as treatment for many diseases. We can remove the genetic diseases. Likewise, so many uses are there. At the same time, there are so many misuses are also there. So now we can see about the points regarding the misuse of genetic engineering. So here you can see some of the points. Threat to indigenous varieties. Indigenous varieties means native varieties. So it is criticized. So some of the people are criticizing that genetically modified varieties cause harm to indigenous varieties and may cause health issues to humans. So there are so many people are criticizing that this genetically modified crops will make many health issues. And so indigenous varieties or native varieties will be disappeared from our earth if we are always promoting the hybrid varieties. Then next is bioweapon, a new challenge. Bioweapon means application of genetically modified pathogen. Disease causing microorganism is known as the pathogen. And pathogens multiplied through biotechnology upon enemies is called the bio war. The applying of this genetically modified pathogens against the enemies is known as the bio war. this becomes a threat to the existence of human beings. So this is a threat to the existence of human beings, bioweapons. Earlier stage, I don't know whether you have heard about the news like this. That means coronavirus was once told that it was used as a bioweapon by China. Like that, certain viruses are used by the certain countries to their enemies in order to destroy those countries. So that's the bioweapons. That means genetically modified pathogens are used against the enemies. Then genetic modification, violation of rights. Certain organizations argue that genetic modification is an intrusion upon freedom of living beings, and it is a violation of rights. So that means God has given us a particular genetic constitution, and we human beings has no right to change that genetic constitution. So some of the people are criticizing the genetic engineering like this. That means it's a violation of rights. Is it clear for you? These are the points about genetic misuse of genetic engineering. I think it's clear for you about the misuse of genetic engineering. So our 7th chapter is over. If you are having any doubt regarding this chapter, we can clear it through the WhatsApp. Read the chapter very well, study very well for the exam. Okay, thank you.